Welcome to section 2 of this unit. We're going to be looking at mitosis and cytokinesis. Hopefully in section 1 you learned about the cell cycle. You learned about gap 1 where it's just normally growing and everything's working great, normal functioning. We talked about synthesis. That's the stage where DNA is duplicated. Okay, it's made an identical copy of itself, and that's going to come into play just here in a minute with mitosis as well. That's important. Needs 2 copies. Then we talked about gap 2, and this is just some normal stuff where it has checkpoints. If it's growing correctly, it's going to move into mitosis. If it's not, something's wrong with the cell. Most of the time it'll just kill itself, and it won't duplicate. So anyway, mitosis, we kind of hinted on it in section 1, is the duplication of the genetic material inside that cell. It's making 2 daughter cells, as we're going to come to call them, out of 1 cell. Mitosis is what happens to help us grow from, a little baby we once were, or actually a single cell we once were, up until what we are today. It also helps repair tissues as we, have injury and normal turnover. Cells and tissues, they die, they flake off, they, do their job and then they're gone. But we need more of them to take their place. So mitosis helps that. First, setting the stage is I want to talk about something called chromosomes. We have this genetic material found inside the nucleus of every cell of our body. If it's a living cell, it has that DNA inside of it. Okay, if we're talking about whatever organism we're talking about, every one of its cells has the exact same DNA inside of it. Chromosomes are this one long continuous thread of DNA. This little X is what you might have seen before. This is just kind of a diagram that represents a chromosome. There's actually 46 chromosomes inside humans. So you have 46 if everything genetically is the norm. You have 46 chromosomes. So, but it always don't look like this X. Most of the time, your DNA is just kind of wound up, especially during like gap 1. It's just wound up like a big wad of spaghetti. All right, these DNA strands are just kind of grouped together, joined together. But we're going to see today that they kind of start pairing up like this because things are very organized. All right, so a chromosome is one longest continuous thread of DNA. But it must be condensed. Condensed means tightly packed. Okay, it needs to be wound up in order to fit into a cell nucleus, especially when we're going to make a new cell nucleus through mitosis. And how it does that is this DNA, the spaghetti strands, wraps around these proteins called histones. When it's loosely condensed, it starts to be called something called chromatin. Chromatin, it doesn't look like those little X's quite so much, but it starts grouping up together around histones. It's kind of like a more organized spaghetti, if you will. So chromatin. Eventually, it will be tightly condensed and packed up together to look like this little X. And these X's are not actually combined. They're not actually conjoined. Each half is an identical set of DNA called a chromatid. Remember, during synthesis or S phase of the cell cycle, say this one, for example, this one side duplicates itself, and it duplicates itself right here. So these are identical copies. Okay, identical copies. We call these sister chromatids. Sister chromatids. All right, so side here and side here are exactly identical. It's setting the stage for in a minute in mitosis when they're going to split apart. And that way we're able to get 2 cells that have identical copies. And it does that because in synthesis, or S phase of the cell cycle, it's made that an identical copy. They're held together by this middle portion here called a centromere. Centromeres, as we'll see, they're not conjoined like by the DNA. It's just kind of held together there. These are easily separated, and thank goodness. So, the end parts, how this DNA strands don't just unwind and uncoil, because it is kind of just grouped up spaghetti, if you will, are called telomeres. Telomeres are found at the end of DNA molecules, and they prevent it from coming apart. It keeps it condensed. It keeps it tightly packed for this process of mitosis. So what is our end game? What are we looking to do? Produce two genetically identical, what we call daughter cells. Doesn't matter if you're male or female, they're just called daughter cells. This is a good little diagram that represents what we're looking for. You see right here at the top, this is a normal everyday cell. It goes through several processes that we're going to learn the names of here in a minute. And in the end, when my toolbar goes away, we have two identical copies that look exactly like the original. Okay, so it looks exactly like the original. So think about, if this is the first cell of a living organism, how do we get to, the trillions of cells that we have now in our body? One step at a time. Mitosis after mitosis after mitosis taking place. Okay, and we're going to learn about this phase right now. Remember, the interphase is the important role in preparing the cell to divide. That interphase makes up of gap 1 synthesis and gap 2. DNA is synthesized or copied, so it has two copies of itself. We see that they're condensed into these things called chromosomes. And now we're going to talk about the nuclei, especially the nuclei, dividing and making two genetically identical nuclei, each with its own single full set of DNA that are the exact same copy. So let's do it. Mitosis. There are four main phases of mitosis. They are very easy to remember with the, I guess it's an anagram or an acronym, I think it is, where you take the first letter of every word and make a thing. It's PMAT, P-M-A-T, PMAT. Okay, so that'll be something worth putting in your memory. That first one is the P is prophase. Prophase. You see several things happening inside the cell right here. The first is DNA condenses into tightly coiled chromosomes. We see those little X's popping up, don't we? We see those little X's, so it's tightly condensed. Nuclear envelope breaks down, or this nuclear membrane. See these little dotted lines? We can see that that's designating it, showing it's breaking apart. So that nucleus is, the material inside the nucleus is getting ready to escape. Okay, it's going to move out. You might remember from our unit on cell organelles, centrioles. We just barely kind of mentioned them. These centrioles move to the poles or to the sides of the cell, two opposite sides, and they start to form these things called spindle fibers. And these fibers are actually going to connect to the chromosomes and pull them away from each other. We'll see that in a minute. So the prophase, the biggest two things are the DNA condenses and the nuclear membrane or nuclear envelope breaks down. All right? So check this out. So PMAT. So next would be the M. Metaphase. Metaphase. In metaphase, spindle fibers that were coming from these centrioles. What is the name of this structure? Of course, those are centrioles. Just wanted to kind of remind you. These spindle fibers attach to the chromosomes. Okay? And what they start to do is they maneuver these chromosomes to the middle or the equator of the cell. And they start to line up across the middle. All right, you can see the nuclear membrane is completely gone by this point. It is no more, and now these chromosomes are kind of free floating. The spindle fibers attach to these chromosomes, and they line them up across the middle of the cell. I like to remember that middle starts with an M. Metaphase starts with M as well. So in metaphase, they're lined up across the middle. No matter what kind of diagram you see, there's several on your page, on your notes. I put them to the left there. I kind of gave you some diagrams instead of making you draw them or anything like that this time on your notes. I wanted you to see them. You can see that it may look a little different from mine, but these chromosomes are lined up across the middle of the cell. All right, PMAT, next is the A, and that is anaphase. Anaphase, all right? The chromatids, we had those chromosomes. Let's go back real quick and see how they were there. There's one chromatid. There's one chromatid. They're attached with that centromere, right? And in anaphase, those spindle fibers actually pull the chromatids apart, moving them to opposite sides of the cell. So they were in the middle. The centrioles, with the spindle fibers' assistance, grabs on and bloop, breaks them apart at the centromere. And they move them apart to opposite sides of the cell. I remember this. Anaphase starts with an A. These are moving the chromatids apart from each other, right? It breaks them apart and moves them across the cell from each other. Okay, so you can already start to see there's something called a cleavage furrow. I don't really like it looking at an anaphase, but it does start to form. We can see the beginning of two new cells. hopefully we're going to see at the end of this mitosis. So PMAT. The next one would be telophase. The fourth and final phase of mitosis is telophase. Okay, there's the T, PMAT. In this one, this cleavage furrow really starts to form. This is where I normally like to designate it. I think your notes has that on there. It starts to bow in a little bit. Nuclear membranes or the nuclear envelopes start to form back. So we have these chromatids that had been pulled apart, right, by these centrioles. Bloop, bloop, bloop. And what begins to happen? The nuclear membrane comes back. Chromosomes begin to uncoil, and the spindle fibers, they fall apart. They're not needed anymore. They're going to flake away, be dissolved into the cell. So look what we have here. We have almost two identical cells, right? These chromatids were exact copies of themselves made during synthesis of the cell cycle. It's moved on into this mitosis, and now through PMAT, prophase, metaphase, anaphase, and telophase, we've got two daughter cells beginning to form. So membrane comes back, chromosomes begin to uncoil. Then there's this process that's not part of mitosis exactly, but we always talk about them together, mitosis and cytokinesis. Cytokinesis is where that cleavage furrow, those two daughter cells are beginning to form right here. It's starting to pinch in. It pinches the rest of the way. Bloop. Remember that cell membrane? We talked about it being very fluid, how it can kind of maneuver like a liquid. And here it separates. Division of the cytoplasm. Cytoplasm is that jelly-like substance filling up the cell. It completely divides. This is a different little process in animal and plant cells because mainly of the cell wall has to be broken down. But for our purposes, just know that cytokinesis is the division of the cytoplasm, giving us two identical daughter cells. Two identical daughter cells. And that's the end of the notes today. But I want you to think about what we have setting up here. We have two identical cells. Now this cell, let's say this one right here, will go through the cell cycle again. It'll go into gap one. It'll grow, it'll flourish, it'll do great. During synthesis, these chromatids will actually duplicate themselves, right? They'll synthesize an exact copy, right? So gap two, all the checkpoints, everything looks great. It starts moving into mitosis again. It's able to break down. These start condensing during prophase, right, forming those chromosomes that are held together by a centromere. The nuclear membrane starts to break down. They line up across the middle in metaphase. Anaphase, they split apart. Telophase, we start to see it starting to pinch in, as well as two nuclei starting to form. And before long, boom, this one has turned into two cells. And the process goes on and on and on and on as needed. Okay, so if we're growing from a one-cell organism at conception, at birth, then we start forming up this process pretty regularly, right? Once a organism becomes mature, like a human becomes mature and fully grown, I guess you could say, this process mainly just happens as we need new cells for repair, whether that's our skin shedding every few days or internal organs that may take years to make new ones. So this is the process of mitosis. PMAT. Feel free to make sure you look at those diagrams. You've got some assignments, including a kind of a little drawing assignment you're going to use your computer tools for. Feel free to use these notes once you get them filled in. Go back through. Know the four stages of PMAT, kind of what to expect in each one. That'll be the biggest takeaway from this section. Let me know if you have any questions.